So before we get started, I want to make a couple of announcements. One, as of about noon today, I caught up on all the grading for the class. I'm now behind again because some people did some work this afternoon. Actually, it's just 3 more things. I did actually just, as we were waiting for class to start here, go through and click through on all of the part 1, 2, or 3 assignments that were submitted late. So that if you were doing the work this afternoon and you're waiting to go on to the next part, I went ahead and did all that just now. But yes, I finally caught up with grading. Also, next week we don't have class. Monday is Patriots Day. It's a holiday. The school is closed, basically. The way that the school makes up for missing Monday holidays, because there's President's Day back in February and then Patriots Day now. To make up for those days, the school will sometimes change the scheduling. And so next week, with Monday being a holiday, the Monday night classes will be meeting on Tuesday, and Tuesday night classes are canceled. So we do not have class next week. If you have a Monday night class, then that meets on Tuesday night. The schedule's not the same for day classes, which is an endless source of frustration for students. I kind of understand why it's this way, and I don't understand why somebody hasn't just gone ahead and fixed it. I actually tried to get something done about it several years ago, and they didn't really follow up on it. But so Monday is a holiday. Day classes make up for that lost Monday on a Wednesday. Okay, so just so everybody's clear, Monday school's closed. Tuesday during the day follows Tuesday classes. At night, Monday classes. Then on Wednesday, the day classes follow a Monday schedule, and then the Wednesday night classes meet as normal. So I just want to be sure that everybody's heard that. It doesn't make any less confusing, I'm sure, but you've heard that's the way that the schedule's set up. It's been true since the beginning of the semester. We've known all along that following Patriots Day, which is next Monday, Monday classes will meet on Tuesday the 21st. So that's been the case since the beginning of the semester. I'm sure you're not necessarily upset about that, but I want to make sure everybody's aware of this. I will not be here. My earlier class gets out about 4 o'clock, so I'm long gone by the time 5:30 rolls around next week. If you show up, I'm not going to be here, so don't bother coming. I think that's all that I needed to mention. Any outstanding issues or questions people have before I go on with stuff? I think we're all set here. So today we're looking at the digestive system. Included in this, I also want to hopefully get to talk about metabolism a little bit, which is actually its own separate chapter. I don't want to talk about all of metabolism. I just want to talk about how carbohydrates are metabolized to make ATP, which goes back to the cellular respiration reaction that I talked about last week. So we'll look at that a little bit more closely. And that's all in the only thing in metabolism I want to talk about. The chapter assignments that you have for this week are based on the digestive system questions. Digestive system chapter. There might be a question or two mixed in with the part 1 and 2 multiple choice stuff that's about metabolism, but the focus is really on the digestive system stuff. Now, the digestive system stuff is actually a little bit dense. It'll take a little bit of time to get through everything. In a class like this, what I want to do to sort of limit the amount of stuff that I'm going to go through is I won't really deal with the anatomy too much. In the lab material, the lab quiz will have some questions about the anatomy and that sort of thing. I'm not going to go through that separately. Instead, what I want to do is I want to move through the system and consider the functions of things. And so as I'm talking about the various things, I'll talk a little bit about the anatomy that goes with it, but I won't talk about it separately. I will mention one thing separately at the beginning just to point it out to you so you know that it's there and you need to pay attention to it. But I'm not going to spend too much time on the anatomy itself. Actually, what I want to start with covering is more of the broader functional aspects of things. So when I go to this presentation I have about the digestive system, did any of you go to the YouTube channel to watch any of the captured stuff that I've been doing? No?
It's okay.
Okay, so some people do. I don't remember if it necessarily has happened with any of my night classes, but I know during the day, definitely, sometimes the video part drops out and there's just that little circular picture of me in the middle of the screen. Part of why I think that's happening is because I'm doing this stuff. The way I have the capture set up, or the way I had the capture set up, I was telling it to just capture what was in my browser window. And so when I went to launch this, it launched into a Adobe Flash animation, which I thought was, I mean, it technically is run within the browser, but it's not run within the browser window. And so when I did that, when I launched things, the video part just disappeared for the rest of the night. So I'm capturing the entire screen, which I don't know if you noticed, so when I pushed the button to start, the screen was one of those infinite regresses. It like had the window that I was clicking on had a picture of the window I was clicking on with a picture of the window I was clicking on with a picture. And it's kind of cool if you go for that kind of infinite regress kind of thing. But it was there. So hopefully this will be captured. If not, we'll see. The splash screen I have here, I kind of wish I had an attribution for it. It's kind of a cool little, funny little picture. It's actually looking at more than just the digestive system. And if you consider what's down here, that's urinary system stuff in there too. But it's a funny little cartoon. What I really want to get into using the presentation here to begin with is to sort of look at the overall function of the system. And what I'm going to do, I'm going to talk more about the function of digestion itself. So if you're taking notes here, you might want to write this list out, and then I'm going to go into digestion in a little bit more detail, so you can put that in afterwards. I'm not going to go into any more detail about the other 4 things in this list, other than just to say that they're here and say a few things about them right now. So when I get to talking about digestion, you probably can't quite read it, but on the screen there next to the word digestion, there are 2 smaller words. So I want to zoom in and look at 2 different aspects of digestion. One of them is mechanical, the other one's chemical. I'll explain what that means when I get into that in more detail. And then the digestive organs are also responsible for propulsion, meaning pushing food through the system. And depending on exactly where we're looking, there's slightly different ways that happens, but it's to some degree based on similar properties throughout the entire system. And then secretion is happening most everywhere. Different amounts of things are being secreted, and some of that I'm actually going to talk about here in terms of the digestion stuff. And then there's absorption. The majority of absorption is happening within the small intestine, although there is a little bit of absorption in the stomach and the large intestine too. And then elimination would be getting rid of everything that we can't digest and make use of. And that's, of course, specifically a function in the large intestine. So that's a overview of all of the functions. But what I want to do is I want to talk about digestion in a little bit more detail. There's two aspects of digestion, mechanical and chemical. Mechanical basically means how we break the food down into smaller and smaller chunks. And then chemical is how we get at the macromolecules in the food and break them apart to get to the nutrient molecules we really want to get to. Now, in mechanical digestion, there's basically three aspects to that. The most obvious aspect of mechanical digestion, which most everybody's probably familiar with, is chewing, because that's the first thing you do. So imagine you're eating a cheeseburger, or if you're a vegetarian, a veggie burger. And what that food looks like before you're eating it is very different from what it's going to end up being like in your body and how you're going to get to break it down. So you have to mechanically digest it down, mix the various components of that cheeseburger, if it were, together. And in the process of chewing, you're going to end up sort of getting it into this sort of pasty state of consistency. And then, and while that's happening in the mouth, there are some enzymes that are going to do a little bit of the chemical digestion of what's going on. Then the food's going to end up getting down to the stomach. And the stomach has its own sort of aspect of mechanical digestion, which is called liquefaction. Or that's the term I'm using for it here. The food that you swallow is kind of a dense, pasty material. When it gets down to the stomach, it's going to mix in with the very watery solution that's in the stomach, and it's going to end up getting mixed in there. And when we get to that part, I actually have a pretty good representation of what that's about in a little while. And then propulsion is actually the next thing on the list after digestion, meaning moving the food along. But in doing that, the food's going to get mechanically changed a little bit. The propulsion works kind of like squeezing toothpaste through a tube. And as that happens, whatever's in the tube is going to get kind of mashed together and that sort of thing. I kind of think about, I guess it's still out there, Aquafresh toothpaste. The way they put it in the tube, there's a red and a blue and white stripes or whatever it is. And when you squeeze it, it's supposed to sort of maintain those stripes in there. But I would imagine, I don't use Aquafresh myself, eventually all the colors kind of get mashed together, and at the end you get this sort of colorful gloop coming out the end. But as things get squeezed through the tubes that make up your digestive tract, they are also going to get physically mashed together a little bit as that's happening. Okay. Now, in chemical digestion, there's sort of three levels to this. And actually, when I was doing this the other day, I realized I really ought to do this in a slightly different order. So let me talk about solvation first, because that's the easiest aspect of this. And that means that things are going to be dissolved. There are some things that you ingest which only take dissolving in water to get them to the state where we use them. The best example of that is sodium chloride, table salt. We need sodium ions and we need chloride ions in our system, and so we get those from NaCl. And if that's on your food, if you're eating a salted pretzel or something like that, as soon as the salt hits your saliva, it's going to start dissolving into the water of your saliva. And you're going to have sodium ions, you're going to have chloride ions. That's all that's necessary to get it to the state you need in your body. So solvation is the most simple aspect of chemical digestion. Now, I want to actually talk about emulsification next, and then I'll get back to enzymes. I have a lot more to say about enzymes, and I'm going to move off of this part of the presentation. There's some other pictures to go with it. So let me talk about emulsification. The term emulsification refers to mixing hydrophobic and hydrophilic components together, or more specifically in terms of food, mixing lipids in with water solutions. Now, we've all had experience with emulsification outside of, what's going on in your digestive tract, but in preparing food, emulsification is necessary also. The simplest example of that would just be shaking up oil and vinegar dressing. So in the bottle, oil and vinegar dressing separates into the oil phase and the vinegar phase. But just picking up the bottle and shaking it is going to make those two phases mix together. And you'll have little oil droplets suspended in the water phase of the vinegar. And then when you pour it out, you have a nice mix of the oil and vinegar on your salad. And then when you put the bottle down, they'll separate back into those two phases simply because lipids are less dense than water, and so the lipids float on top of the water. That's just a simple physical emulsification of that oil and water mixture, but the same sort of thing happens in other situations. And really, if we kind of expand what we think about of salad dressing, we can see a pretty good example of a less transient emulsification. So you might not like oil and vinegar dressing. You might like something else. What are other types of salad dressings besides oil and vinegar? A vinaigrette's basically an oil and vinegar dressing, right?
I said Italian.
So in Italian, that's also oil and vinegar. Come on, we've got some more. French. There we go. What else?
Ranch.
Ranch. Ranch. Ranch.
Blue cheese.
Blue cheese. Thank you. What's the basis of these types of salad dressings? Anybody know?
Dairy.
Not really.
Mayonnaise.
Mayonnaise, right. What is mayonnaise?
Olive oil and egg.
Eggs and oil. Olive oils, using the make mess. Oil obviously is a lipid phase. Eggs are, or at least the white part of eggs, are a water solution. It's the protein albumin suspended in water. It's a rather thick suspension, but it's still a water suspension. When you whisk together eggs and oil, it becomes a homogenized, emulsified mixture. The emulsifying agent is in fact something in eggs. Right? And a lot of things in cooking that need emulsification, you just add an egg to it and emulsifies it. If you're making a cake batter, you've got butter or whatever lipid source in there, and you've got the flour dissolved in some sort of water solution. To mix those together, you add an egg, and the emulsifying agent in the egg does that. What emulsifies things in the egg is a substance called lecithin, which is found in other food sources too, but in egg, that's a pretty good source of it, especially for baking. I think lecithin is found along the outside of the yolk, because the yolk is going to have some lipids to it. So I think the lecithin sort of helps with the separating out the yolk part from the white part of the egg. I'm not positive about that, but I think that's what lecithin does in the egg. It's not the egg came along because it co-evolved with humans' baking culture or something like that. The egg just got better and better at helping us make cake batters. Come on, that's the jokes people laughing. Emulsification in your body works on a similar principle. There's a certain type of molecule that's able to get in and help lipids combine with water solutions so that they're suspended within that. And the digestive juice that does that for us is called bile. It contains within it a class of molecules called bile salts, which get in and break lipids apart. And when the lipids are broken apart, so they aren't forming oil droplets within the greater mixture of foods that you're eating, then the enzymes that are going to break them down can get in and do their job. Now, you're already familiar with one of the bile salts, one of the more common bile salts, and that's bilirubin. Where did you learn about bilirubin already? I'm expecting silence here because people don't usually pick up on it.
I know it's, I know this because I have a high count of it, but I gotta check with blood tests. I believe it's something with the liver.
So bile's made by the liver, that's indeed correct. But probably from the clinical aspect of it, you're not really caring about where the bilirubin originally came from.
Don't they look for it in urine as well?
So eventually bilirubin becomes your bilinogen, which is found in the urine. But that's the wrong direction. We want to go back to its source. Anybody remember where it came from? It's actually a repurposing of the heme molecule. When red blood cells live out their 120-day lifespan, they're broken down by macrophages in the liver or the spleen or the bone marrow. And most of the cell is just recycled to make more cellular components. The iron ion is picked up by transferrin, which then goes to be used to make more hemoglobin for red blood cells. The heme molecule, however, after the iron's been removed from it, ends up going to the liver where it's changed into an intermediate molecule and then finally into bilirubin, and it's used as a bile salt. Initially, one of the bile salts that we have started its life as the heme molecule in hemoglobin in red blood cells. And then it just gets repurposed as a bile salt later on. But what it does is it basically works its way into lipid molecules and helps kind of separate them apart so that the enzymes can get in there and break the covalent bonds that hold those molecules together. So I want to talk now about enzymes. I said I want to skip over that and do that last because I want to talk a little bit more about enzymes than just what's referring to the word in this list. This table is from the book, which just runs through a number of different enzymes. The first column here says enzyme category. So we have salivary, gastric, brush border, and pancreatic enzymes. Salivary means they're in the saliva. Gastric means they're made in the stomach. Brush border is referring to things that are found along the inner surface of the small intestine, which is called the brush border. And then the pancreas makes enzymes which get into the small intestine also, and they're not found at the edge of the small intestine, but they mix in with the food as it's passing through the small intestine. So those are the different types of enzymes. I want to talk about a few of them. And what I want to start talking about is there are two right here, but there's actually a third that for some reason got left off of this list, which is strange because it's actually sort of a more straightforward example of that kind of enzyme. So, oh, good, yeah. This is a similar table. It's just out of a different book, and the reason I'm grabbing it is because because it has what I want to talk about right here. So in the table from our book that I was just showing you, it has listed two of these three right here, and I just want to mention the third one also. So I'm going to drop out of the presentation for a second so I can start writing this stuff up on the board. So we have Sucrase, maltase, and lactase. These three enzymes are all specifically breaking disaccharides down into monosaccharides. And there are three main disaccharides that we deal with made up of some combination of the three main monosaccharides. Sucrose is a glucose molecule and a fructose molecule that are bound together. And the bond between them is broken. Oh, sorry. Getting ahead of myself there. Sucrase is the enzyme that breaks apart this molecule, which is called sucrose. OSE ending means that it's a sugar, ASE meaning that it's an enzyme. So sucrase is the enzyme that breaks down sucrose, which is a glucose molecule and a fructose molecule bound together as a disaccharide. Maltase breaks down maltose, which is 2 glucose molecules bound together. Okay. And then lactase breaks down lactose, which is a glucose molecule and a galactose molecule. 2 simple sugars there. Now, what do we associate sucrose with? Where do we usually find that? Artificial sweeteners are trying to be like sucrose. That's natural sweetener. That's table sugar. Okay. You're probably thinking of sucralose, which is the Splenda artificial sweetener. What's the main agricultural source of sucrose? It is sugar, yes. Where do we get it from? What? Sugar cane is actually not the main source, sort of the second most popular source, because it only grows in certain climates. Nobody ever knows this. Sugar beets. Ever heard of sugar beets? They grow in a wider temperate zone, I guess you would say, so we can grow more of them. And they're usually a better source of that. Where do we get maltose from? It's more often referred to as malt. What's a source of malt? Or what do you usually associate the word malt with?
Beer.
Beer, malt beverages. Okay. What do you make beer from?
Yeast.
Grains, not the yeast. The yeast eats the maltose from the grain. So barley, rye, those sorts of things are sources of maltose. And then the yeast breaks maltose down for its own food. And in eating the glucose molecules that it makes out of breaking that down, it gives off ethanol as a waste product. And that's what makes the beer alcoholic. Where does lactose come from? Yeah, it's milk sugar. Now, the simple sugar, the monosaccharide galactose, I'm abbreviating it GAL here. The full word would just be lactose with a GA on the front of it. So lactose is the name for the disaccharide we find in sugar, which is made up of glucose and galactose, which is the milk sugar specifically. Now, of course, all sugars are made by plants through photosynthesis, but mammals, when they're producing milk for their young, are going to take a simple sugar, maybe like glucose, and modify it to be a slightly different arrangement of atoms, which we call galactose. We find that in milk. Now, what word does galactose remind you of? Galaxy, right. What galaxy do we live in? The Milky Way galaxy. It's not a coincidence. The Greek word that gives us galaxy and milk, there's one common word for that. So some Greek guy back in the past looked up at the sky and saw this big spill of stars across the sky and said, oh, it looks like spilled milk. So it got called the Milky Way galaxy. Actually, he didn't say, hey, I'm going to name this after my chocolate bar or something like that. Just the word for galaxy and the word for milk are the same because the Milky Way, the stars of the Milky Way galaxy in the sky kind of look like spilled milk across the sky. That's kind of where it comes from. Just a little interesting factoid of what the words mean that I kind of enjoy. These enzymes are breaking down these disaccharides to get the monosaccharides out of them. These are found at the edge of this small intestine lining, at the brush border, because it's the last enzymatic step to get that macromolecule ready to be absorbed, we absorb the simple sugars. We don't absorb larger sugars. But we don't necessarily only eat disaccharides. We eat complex carbohydrates also. And in the table here that I'm using from a different book, the other carbohydrate enzymes that are mentioned are both amylases. One's found in the saliva and the other's found in the pancreas. Amylase breaks down complex carbohydrates like starch. Amyl actually means starch, so it's the enzyme that breaks down starch. It's going to break them down, a big chain of sugar molecules will get broken down into disaccharides and trisaccharides, and then maltose, sucrose, and lactose can break those down. And really, maltase breaks down bonds between two glucose molecules. Sucrase breaks bonds between a glucose and a fructose molecule. And lactase breaks bonds between a glucose and a galactose molecule. If I said ose instead of ace anywhere in there, or ace instead of ose, I always get those mixed up as I'm talking. But these enzymes break bonds that we would see in those disaccharides. So actually they can take some of those trisaccharides and break them down because they work on that bond. Really, all of the disaccharides have one glucose molecule, and so the different enzymes are really just based on what the second sugar in the disaccharide is. So maltose takes a glucose off of a glucose, sucrase takes a fructose off of a glucose, and lactase takes a galactose off of a glucose. So there's that. The next thing I want to talk about, actually, I want to refer to this picture a little bit. This is an image from the science publication Nature, which the source is down here. And it was an archaeology study looking at the revolution of milk or dairy agriculture. Whoops. And this figure from the book is just looking at the prevalence of lactose tolerance, meaning adults able to break down the lactose disaccharide associated with the geographical location that person came from. I've not read this particular paper in detail. I just sort of skimmed the abstract to get an idea of what's going on with it. So I don't remember anymore now if they were in fact sampling populations in these geographical locations or just sampling populations and based on their genetic makeup, assigning them to some ancestral population. And so as the map gets to darker colors, it's basically showing where people developed a genetic ability to continue breaking down lactose as adults. Now, everybody should be able to break down lactose as a baby because that's your only form of nutrition. We're mammals. That's the whole point of being a mammal, is that the young are fed through mother's milk. Now, there are certainly exceptions to that. Some people can be born with complications where they don't handle lactose as a child, and that can be a pretty severe problem. But we have the ability to deal with that thanks to modern medical technology. But putting those exceptions aside, everybody should be able to break down lactose as a baby, and then as an adult, that capability tends to go away, except for the places where we see the dark spots here. And what we see is Northern Europe has a real dark spot, and then a few places along the equator, including the Saudi Peninsula and the eastern part of the Indian subcontinent, and then far western Africa. Yeah, western. Wait, I said that wrong. The western part of India and far western Africa. What this archaeological study was getting at, and some other data that's not represented in this picture, was considering evidence of dairy agriculture in those locations. And what they found was looking at what's on the inside surface of clay pots dug up from locations around there, they could say how prevalent dairy agriculture was. Just that milk and other dairy products would be stored within a clay pot. Over the years, there'd be some molecular evidence of the use of those clay pots. So we could see when dairy agriculture was prevalent and where it was prevalent in different areas. And so what they're seeing here is that dairy agriculture seems to correspond to these lactose tolerance hotspots. Those equatorial regions probably correspond mostly to, more in the Saudi and African regions, to goat dairy agriculture. I'm not that familiar with Indian agriculture. I don't know if they're more goat or cow oriented. I think goat, but I don't want to make any assumptions because I really don't know. But I do know Northern Europe dairy agriculture was mostly cow. And so a lot of immigrants to the eastern United States during the 16 to 1800s were coming from Northern Europe. So lactose tolerance tends to be fairly common in America, which Ben and Jerry's loves to hear, because of most of the ancestral sources of people coming over. When you see people that are lactose intolerant, you can usually trace it back to that they might be from one of these other places. So we usually associate far eastern ancestral stock, so to speak, with being lactose intolerant, that sort of thing. And some of the southern European locales. So this picture kind of, or the data from the study kind of gets at that there might be a direct tie-in between the genetic ability for somebody to break down lactose as adults with their original ancestral location. This study, which came out in 2013, so it's a couple of years old now, their main interpretation of the results was basically that in these locations, back when dairy agriculture was common, or where it was starting, which I think was 8,000 to 6,000 years ago or something like that, individuals that were by chance able to break down lactose as adults would be able to survive better and pass that genetic ability on because they could get more energy out of the food they ate using dairy agriculture. So there seems to be some sort of suggestion of a link between the prominence of the genetic trait of being lactose tolerant with locations where dairy agriculture started out. Like I said, this is about 2 years old. I just read a study earlier this year that, or not a study, but a news article referring to some studies that I didn't read, that actually suggests that maybe what this paper was saying wasn't entirely true. There might be some suggestion that a lot of the modern traits we associate with humans, whether it's lactose tolerance, and actually this article I was reading was talking more about the Northern European thing, so it was also talking about lighter skin color, that those traits probably didn't really show up until 25 or 3,500 years ago. So the archaeological evidence of dairy agriculture goes back to like 6,000 or 8,000 years ago, but the genetic evidence of these traits, like lactose tolerance, doesn't quite match up with that. The fact that there is dairy agriculture in certain areas and there might not have been lactose tolerance in the adults at that point is pretty easy to resolve, because in dairy agriculture, an easy way to make an adult able to eat dairy is to just have something else break down lactose. Okay? And the easiest way to do that is to use microorganisms. In a lot of these places where dairy agriculture is found, yogurt is a common component of that dairy agriculture, because you can take lactose-rich milk products from goat or cow or whatever it might be, if you allow that to be fermented by microorganisms like bacteria, those microorganisms will break down the lactose. And then when a human eats it, they're eating the monosaccharides that come from the microbes' work on that lactose. So that might suggest why the agriculture existed far before the genetics did. And then the genetics might have come along because there was an advantage to people not having to necessarily eat something that's already been processed by microorganisms, which kind of gets to the question of when do you know that yogurt's gone bad? I mean, it doesn't smell that great, and it's got bacteria in it, so isn't that kind of how you describe spoiled food anyways? So if you didn't have to rely on microorganisms to do the part of the digestion for you, you're more likely to survive and pass on the genetic traits or something like that. But anyways, I kind of like to include this in talking about the enzymes to get you thinking a little bit broader about things than just the basic A and P. There are, cultural or at least agricultural ramifications of what's going on within the variation that we see in the human population. We usually think of lactose intolerant people as being weird because they can't eat Ben and Jerry's or whatever. But really, that's the normal situation. People who are lactose tolerant are the weird ones. Okay? And it's really limited to a few geographical locations where that trait came from. So don't laugh at somebody because they can't eat dairy. That's the norm for humans. And being able to digest lactose as an adult is actually kind of unusual. It's just very common in the United States because a lot of the ancestral origins of the United States come from regions where lactose tolerance was common. Okay? So it's kind of interesting to think about the broader implications of what's going on with the system there. Now, I'm not going to talk about all these other enzymes. I just want to point out some things. Up above the amylase and the sucrase, maltase, and lactase, those were breaking down carbohydrates. These are all enzymes that break down proteins, what we call proteases collectively as a group. I do want to point out a couple here. First off is pepsin. The reason I want to point this out is that this is the main enzyme that we find in the stomach gastric juice. And it's just a general protease. It breaks down proteins. So when proteins get to your stomach, you start to chemically digest them. And I'll mention that when we're talking about stomach function a little bit more. The other one I want to point out here is renin, R-E-N-N-I-N. Now, there's two reasons I want to point this out. One is I don't want you to confuse that with another molecule that we'll talk about in the urinary system that's also called renin. This one is R-E-N-N-I-N. The one we're going to meet with the kidneys is R-E-N-I-N. Now, the other renin really should be pronounced renin, but nobody pronounces it that way. Everybody pronounces it renin. Because the other renin, this one that we're talking about right here, is actually a fairly minor enzyme that anybody runs across. Now, the kidney's renin, really renin, is a hormone, which helps to regulate blood pressure. This renin is an enzyme produced in the stomach, which helps to break down milk proteins. And so that kind of brings me to the other reason I want to talk about it, sort of in keeping with that dairy agriculture thing I was just talking about. Renin, the stomach protein for milk, stomach enzyme for milk proteins, is very important with dairy agriculture because it's an important part of making cheese. Does anybody know how to make cheese?
Yes.
Would you like to tell us quickly how that's done?
Oh, God.
I can tell you in 2 minutes, even 1 minute. Okay. Milk spoils. Put it in a dark, cold place, turns into cheese. Okay. Very basic. Really what it is milk gets an acidic pH. Okay. The proteins in milk start to denature and break down. You let it sit out for a while. The water part of the milk separates from the solid part. If you've ever left your milk in the refrigerator for too long, you know what that looks like. That's what's called curds and whey. Okay, so little Miss Muffet sat on her tuffet eating spoiled milk. Along came a spider, sat down beside her and said, "Ooh, gross." But come on, folks, these are good jokes. Wake up. What? Anyways, that's curds and whey. You separate the whey, the water part, from the curds, the disgusting spoiled milk part. Put that in a cold, dark room for a little while, months, years, who knows how long, and it will turn into a hard cheese. Okay? That's how you make cheese. Now, that's a simple explanation. What renin has to do with it is it's actually part of that initial phase of making the curds, because the milk proteins have to be broken down. That happens in two ways. One is they denature because the pH of the milk becomes acidic, and so the protein structure just changes from its normal three-dimensional structure to something else, and that's what makes the clumpiness in the milk. But then also the proteins have to be broken down a little bit, which just adds to the clumpiness of the milk. I don't know if this is actually how it was discovered, but the story of how making cheese was originally arrived at has to be really strange. So my way of thinking about it is we have a caveman, let's call him Og. And back then, you killed an animal for meat and for fur and for all sorts of other things. You didn't want to waste anything from that animal because it was kind of hard to kill animals when you were a caveman. And so what Og did is he used the various hollow organs like the stomach as pouches to carry things around. Okay? And the stomach is a great pouch for carrying things around because it's pretty sizable and just has an opening at either end. And if you sew off one, or if you close off one opening, and the other opening you can use as a spout. It's a great little thing for carrying things around, sack for carrying things around, like liquids. It's great for drinking out of. I remember when I was a kid, my dad had this wine pouch that he'd carry around when we went skiing, and it was stomach shaped because it was sort of that idea. It was called a wine bladder, which sounds kind of disgusting now to think about it. But, it's basically a stomach shape with one end closed off and the other end available for pouring whatever liquid is out. So we have something like this. So you can close off this end and this becomes a spout that you can drink out of. Okay. So Og has a stomach that he's repurposed from whatever animal that he killed off. Now, what you have to do when you're repurposing organs out of animals, you have to tan I guess you'd call it tanning. You have to treat the tissue. You have to let it become more leather-like to be able to use. So let's say Og is a little bit lazy. Okay? So he decides that he's just going to take the stomach. He's not going to tan it completely, and it's not completely ready to do its job. He puts milk in the stomach, and then he goes marching across. For some reason, Og, in my mind, lives in the Swiss Alps. So he goes marching across the mountains, which is not an easy march to make. At the end of his march, he sits down and he decides he's going to have something to drink. So he gets out his little stomach milk bladder, and he goes to drink it, and it's all this clumpy, spoiled milk. Okay. But again, Og doesn't want to lose anything, so he says, well, what can I do? And he separates the curds from the whey, and he wraps it up in something and sticks it in the bottom of his pouch and keeps walking on. Okay. He puts the pouch back in his cave, wherever he lives, when he gets to it, and forgets about it for a few months. Okay. It's cold, it's dark, it's damp way back in that cave. When he finally gets around to thinking, oh, I put that thing back there, he pulls out the little curds that he wrapped up in that, and he's suddenly got cheddar cheese, or Swiss cheese, I guess, if he's living in the Alps. Okay. That's how I picture cheese making coming to be in the first place. Og was just lazy, and he left some cheese curds in something in the back of his cave for a long time, and it became cheese. Okay. I don't know if that's true, but I kind of like that story. Again, kind of introducing you to some other aspects of what feeds into, no pun intended, the digestive system stuff. Okay. So in Og's story, he didn't really prepare that stomach completely. So it still had a little bit of the renin left over from it. And so that rennin helped to spoil the milk, or at least break down the milk protein so that it became curds and he could make his cheese from that. Now, actually, if you're making cheese and you want to do all of this, if you take the curds and you add salt to it, that's cottage cheese. Okay. I don't suggest you do that. Go get some spoiled milk out of your fridge and add salt to it and eat it. But that's what cottage cheese is. And then if you just put that cottage cheese in a cold, dark place with a few microorganisms for a while, it will turn into hard cheese as more and more of the moisture comes out of it. So. Anyway, so those are proteases, things that break down proteins. And then the next group of enzymes are called lipases. They break down lipids. The table from our book that I showed you earlier has exactly the same information. Well, not exactly the same information, but points out these two lipases too. And I'd just like to take a second because I don't think it's entirely correct. Most every A&P book is going to mention that there are these two lipases. Actually, there's some of them that will mention 3 of them. But of these 2, I don't really believe lingual lipase exists. And I want to tell you why, because it does give a little bit of a picture of what's going on in the digestive system, especially in the mouth. Humans have a sort of inborn need to find carbohydrates in our food. It is the sole energy source for our central nervous system. Okay. Now, mitochondria can make ATP from carbohydrates, lipids, or amino acids. But in your brain, they can't, because lipids are used for myelin and proteins are used for ion channels and neurotransmitter receptors and cytoskeletal components and all that sort of thing. They're needed for other structural components within the nervous system. So the nervous system only uses carbohydrates for energy. When you eat something, the salivary amylase will start to break down whatever carbohydrates are present. And that should release some individual glucose molecules from whatever you're eating. The longer something with complex carbohydrates stays in your mouth, the more that will happen. Okay? If you take a bite of a bagel, plain bagel, so it doesn't have raisins or cinnamon or anything like that in it, take a bite of a bagel, you eat it real quick and you swallow it, it won't taste that sweet because you're not really processing the starches in it that much at all. But if you get some bagel in your mouth and you sort of leave it in your mouth for a while, let it sort of digest within the saliva, it'll start to be a little bit more sweet tasting because glucose within the complex carbohydrates ends up getting liberated and binds to the glucose receptors on your tongue. Okay. So if you're Og the caveman again, and you're trying to find food, you taste the food first, and if it tastes a little bit sweet, then it's a good source of carbohydrates, and your body's going to want that. We have an ingrained reaction to sweet-tasting food that when we taste sweet food, we want more of it. Because if Og is getting carbohydrates in his food, he wants to get as much as possible into his system that he can. Okay. Nowadays, we don't need to do that, because we can just go to the grocery store and get more carbohydrate-based food and have more carbohydrates for power in our brain. But we still have that ingrained reaction to sweet tastes, which is, to some degree, kind of the problem with obesity. If you taste something sweet, you want to eat more. So that's what's going on in the human mouth. Okay, we're really looking for carbohydrates. Carbohydrates. In the book here, they're mentioning, well, this is from a different book, but in our book also, they're mentioning lingual lipase, which I don't really think is true for humans. As I've tried to research information about lingual lipase, what I always come across are 2 things, both of which kind of support my view. One is any sources talking about lingual lipase in this, which is released in the saliva, is talking about the rat model. I've not seen any good sources talking about lingual lipase actually being present in human saliva. Now, rats are different from humans. Okay? Now, there are a lot of things about rats that are similar to humans, and they make a pretty good animal model on a lot of different points. And probably people will think that rats and humans are great that because of similarities between rats and humans, they're great models for digestive behavior. And one of the reasons for that is that humans and rats are both omnivores. We eat both meat and vegetable matter to get energy. The difference is that rats are scavengers and humans are hunter-gatherers. So how we behaviorally get our food is very different from rats. Rats are scavenging, they're looking for the best source of energy that's possible. Also, rats have a much more simple brain structure than we do. So they don't need quite as much energy for their brain to work as we do. So they're looking for the best energy source. And lipids, by far, have more energy in them than carbohydrates. So rats want a lipid-rich diet. We don't want a lipid-rich diet. Right? While it's a great source of energy, it's not a primary energy source for our nervous system. We want the carbohydrates. So I don't think humans have lingual lipase because we really don't need to sample the presence of lipids in our food. Another thing that I think goes with that is a study that came out probably a few years ago now, showing that there are genes for lipid receptors that could be part of the sense of taste, but no evidence that they're actually present in humans. So rats probably have lipid receptors that make for a sixth part of their sense of taste. Humans have those genes, but we just don't use them. Okay? So they're probably present in most animals, but we probably don't use them. There's no evidence that we have a taste for lipids. So if we don't have a taste for lipids, there's no point in having enzymes that break lipids down in the mouth. Okay? Which also goes with the fact that lipids need to be emulsified before we can break them down. And the only emulsification that can possibly happen in our mouth is physical emulsification. So sure, if you put oil and vinegar in your mouth, it'll mix up just fine. Don't suggest it, it probably doesn't taste that great. But when you're mixing a bunch of food together and you're chewing it up, the lack of any kind of chemical, if you will, emulsification means that the lipids within the food that you're eating are going to tend to cluster together into oil droplets within what you're eating. So they're not going to be sufficiently emulsified for a lipase to get them to do anything. The emulsification that we have shows up in the small intestine. So pancreatic lipase is what breaks down lipids in the small intestine. That makes perfect sense. Okay. Now there actually is a third lipase, which isn't for some reason listed here, and I don't think it's listed in our book either, which is called gastric lipase. And part of researching lipases, I've looked into that also. It does in fact exist. The thing is that it's not active until it reaches a neutral pH. So while it's made in the stomach, it's not active. It doesn't break down lipids in the stomach. But as soon as the material from the stomach move on to the small intestine and the pH changes, then the lipase is active and it'll start breaking down lipids. It's already mixed in with them to some degree, so it can be available to help the pancreatic lipase break down fat molecules into smaller lipids in the small intestine. I also want to point out here at the end nucleases. When I talk about nutrient molecules, I'm usually talking about carbohydrates, lipids, and amino acids, or proteins. And I'm basing that largely on what you're probably familiar with looking at food labeling. Those are the things that are listed in the foods that you're eating. There's nothing on food labels that say nucleic acids. But there are nucleic acids in everything that you eat. Just not enough to be nutritionally relevant. Nucleic acids contain in them a ribose or deoxyribose sugar, which would be a carbohydrate kind of thing. And they also contain phosphates and these base molecules. But there's not enough of them to be relevant nutritionally. There's no caloric content associated with them. If you think of an extreme case, well, I'll get to the extreme case in a second. All the things that we eat ultimately come from some sort of living organism source, okay? Whether it's plants or animals or whatever. So there's got to be some sort of cellular material in everything that we eat, which would include the nuclei of cells and therefore some DNA and RNA in the form of ribosomes and that sort of thing. Just not enough to be nutritionally relevant to our food compared to the proteins and lipids and that sort of thing that's mixed in there. An extreme example of that would be an egg. Okay, so an egg has a lot of protein in the white part, which is the albumin, has a lot of lipids in the yolk part, doesn't really have any carbohydrates, and then it has one cell. Okay, an egg is just the support material for one cell that's going to eventually become a chicken. So at the point where you're eating the egg, they're really only eating one cell, and we all know that cells are very small, so there's not a whole lot of DNA in there compared to the protein and lipids that are present. Okay. But we do get nucleic acids in there, so there are nucleases that are made by the pancreas that help break down nucleic acids. Other than mentioning right here, I'm not going to talk about that at all because nutritionally they're not really that relevant to things, but to let you know that it's there. So that kind of wraps up talking about the functions. Like I said, I'm not really going to talk about any of these other functions here. I just wanted to talk about digestion in a little bit of detail because this is the digestive system. And we wanted to think about what digestion really means. Okay. So from there, I want to move to the book. And like I said, I'm not going to really talk about the anatomy in a whole lot of detail, other than it's going to work into what the other things we're talking about are. But here at the beginning, it's setting up what the digestive system is. And this sort of runs through the different structures that make up the digestive system. And there's two groups of organs. There are what the book is referring to as alimentary canal organs, which more appropriately or more commonly is referred to as the digestive tract or the GI tract, gastrointestinal tract. And in fact, right here in the text, they haven't bold-faced it, but the gastrointestinal tract is another name for what's called the alimentary canal. These are all the organs that food actually passes through. They're hollow organs, and they contain, it's what the food passes through as it's being digested and absorbed. And then what's not digested and absorbed at the end is eliminated as feces. The other structures are accessory digestive organs. And these are anything that aid in digestive function. Now in the book here, they specifically define that as anything that aids in the breakdown of food, which is fine. Just sometimes we can kind of expand accessory a little bit, anything that aids in digestive function. When we talk about the accessory structures, we usually keep to the ones that are actually about providing digestive juices. So there's the salivary glands, the liver, gallbladder, and pancreas. Those are the main accessory structures, and we'll talk about those. The other thing about the anatomy that I want to point out, and I don't want to talk about it in a whole lot of detail, has to do with this picture here. And this is a common picture that's going to be in any kind of textbook like this, showing the different layers that make up a digestive organ, a digestive tract organ. There's 4 layers, which are highlighted in boldface here. There's the mucosa, which is the innermost layer, the submucosa, which is adjacent to that, the muscularis, which is the muscular layer adjacent to that, and the serosa, which is covering the outside of the organs. The mucosa includes the epithelium that's absorbing the food, the nutrients that we're eating, as well as its associated connective tissue called the lamina propria, and it has a little bit of muscular tissue associated with it too, okay? Which is called the muscularis mucosae, not to be confused with the muscularis, which is the main muscle structure. Glands of the digestive system connect to the mucosa and release their contents into the lumen of the digestive tract. The lumen of the digestive tract is considered outside your body from the immunological definition I was talking about a few weeks ago now, as well as the endocrine versus exocrine definition. So exocrine glands secrete things outside the body, which in this case includes the lumen of the digestive tract. Some of those glands are within the mucosa. Some of those glands are actually in the submucosa and reach out to the mucosa. And some of those glands are outside the digestive tract completely, like the pancreas, liver, gallbladder, and salivary glands. Now, the gallbladder is not actually a gland. It's just a place that holds the secretions from the liver, but the liver and the gallbladder together connect to the digestive tract through the same duct. There's not a whole lot to say about the submucosa. It's just connective tissue outside the mucosa. The muscularis has, for most locations, two layers, a circular layer and a longitudinal layer. The circular layer are fibers arranged around the circumference of the tube. And when they contract, they pinch in on the tube. And then the longitudinal layer are fibers that run the length of the tube. And when they contract, they basically shorten the tube up in that little region, or they help push things along the length of the tube. Those are very important for propulsion. I just want to mention that in the stomach, there's a third layer, which is at an angle to both of these. The longitudinal and circular fibers are basically at 90 degrees to each other. And then there's a 45-degree angle layer in the stomach called the oblique fibers. And I'll point that out when we get to the stomach. And then the serosa is just the outside covering. Now, this is the digestive system's version of what we saw for the lungs and the heart. Okay, so the pericardium in the heart and the pleura of the lungs are equivalent to the serosa covering of the digestive system. And in the digestive system, its specific name is the peritoneum, which unfortunately isn't in this figure right here. But the peritoneum covers the outside of the digestive tract organs in the abdominal cavity and lines the inside surface of the abdominal cavity. The abdominal cavity can also be called the peritoneal cavity because it's lined with the peritoneum. The distance from an organ to the inner lining of the peritoneum, abdominal cavity, isn't necessarily as close as we'd see for the pleura or the pericardium. So where the serosa extends away from an organ to the body wall, that area is called the mesentery. And within the two layers of the mesentery, we have blood vessels and nerves and lymphatic vessels passing through this. And that's how those structures get out to the particular structure of the organ there. Okay. That's all I really want to mention here. You can get more detail from the reading, but I've pointed out the things that are going to be relevant to what we're talking about. So here's a picture of just a transverse section through the abdominal cavity, and the aqua blue kind of color there highlights where the peritoneum is, where it's covering the outside of digestive organs and where it's lining the inside of the abdominal cavity. Not just the anterior wall, but also the posterior wall. It sort of covers up some of the organs that are in the posterior wall of the body, okay, which we see back here. So that's the end of the anatomy part. There's a section in the book here that gets at the processes and functions, which gets a little bit into the list I showed you there before. The one thing I want to point out, a good picture in this part of the book, this is showing a particular type of propulsion. This is what's called peristalsis. Let me write that word a little bit bigger. This is a propulsion of food through the GI tract. And so this picture here is showing the most simple description of what this is. The circular fibers pinch in behind a chunk of food or a bolus of food, and then the longitudinal fibers kind of help to push that food down the length of the tube. And then more fibers pinch behind it, and more circular fibers pinch behind it, and more longitudinal fibers push it further and further on. This picture kind of depicts what's happening as we're swallowing food down the esophagus, which is where the most simple peristalsis happens. But this basic idea is going to happen in most locations to some degree. That might be a little bit more complicated with other movements going on, but the basis of movement of propulsion really is about peristalsis, which is usually the tube pinching off behind the food, and then it pushes it along, squeezes it through the tube there. Where are we here? Okay. This figure here gets at sort of where propulsion, digestion, absorption, those sorts of things take place, as well as elimination, which is defecation at the end. I'm not crazy about this because they're trying to put all these little icons in to represent the different types of functions, and I don't think they do a great job of putting it everywhere. So, for instance, for mechanical digestion, it has these two little circular arrows, like churning things around, and it mentions chewing in the mouth. But if you look at the mouth, there are no little circular arrows suggesting that's where it's taking place. So it's a little weird representation of that stuff. All I can think is this, instead of circular arrows, it has this sort of zigzag line with an arrow at the end representing swallowing. But it's a kind of weird picture nonetheless. But it does summarize a lot of those functions there. I'm not going to go into the neural and hormonal controls, generally speaking. I will talk about them in terms of the stomach and the small intestines, which is a good example of where that stuff is happening. So I'll get to that. As far as the mouth is concerned, all that I really want to mention here is about swallowing. So it talks about chewing and the production of saliva. There's salivary glands in the sort of at the corner of the cheek, jaw area, as well as underneath the tongue and the jaw here. Those are the salivary glands, which are accessory organs. And it talks about teeth. I don't talk about teeth that much in this class, just because in the interest of time, I'm trying to move past it. If you're planning on going into dental hygiene, I apologize. You can go back and read that on your own in some detail. We already talked about the pharynx to some degree in terms of the respiratory tract: nasopharynx, oropharynx, laryngopharynx. The nasopharynx is not part of the digestive tract, but the other two are. And so as we're swallowing food, it's going to go down that way. Then it's going to end up going into the esophagus. There we go. I was waiting for this picture. This is talking about the process of actually swallowing, which is more formally called deglutition. And this is actually not showing all of the swallowing function, and because rather deglutition is more talking about the reflexive part of it. Swallowing actually comes in two phases. There's the voluntary and the involuntary parts of it. The voluntary part is really about you moving food in your mouth around with your tongue towards the back of the mouth. If you want to swallow something, you push stuff back towards the back of your mouth. Okay? And that's done voluntarily. If you don't want to swallow something, sorry, I'm thirsty. I wanted to demonstrate that way. But you don't have to actively swallow something if you don't want to, because it depends on when you push it towards the back of your mouth. Once it hits the back of your mouth, it's going to start setting off the swallowing reflexes, which will be the soft palate moves back across the throat, blocking off the nasopharynx so food doesn't go up. Not a perfect system. If you've ever seen a kid spit milk out of its nose, you know there's a problem there. But it's supposed to work that way so that food goes down. And there are some constrictor muscles in the pharynx which will help to move the food down. When it gets down to the laryngopharynx, the epiglottis will cover up the lower respiratory tract so that the food goes towards the esophagus. And then when it gets to the esophagus, that picture we were looking at a second ago with peristalsis is basically what happens, pushing the food down the rest of the way, okay, until it gets to the stomach. So that's essentially what's happening in swallowing, and they break it up into the voluntary and the involuntary parts. Involuntary actually happens partly in the pharynx and partly in the esophagus, which eventually gets it to the stomach, okay, which is the next subject here. Now, I mentioned when I was talking about the structure of digestive tract organs that the stomach actually has a third layer of muscle. It has circular fibers and it has longitudinal fibers, but it also has what we call oblique fibers that are at an angle. The circular and longitudinal fibers are basically 90 degrees relative to each other, and so the oblique fibers are kind of 45 degrees to either of those, kind of in between them. Sort of like circular fibers are going around this wall of the stomach that way, longitudinal fibers are going this way, oblique fibers are going to go across in this direction. And it just allows for the stomach to do more movements than, say, the esophagus or the intestines. Because what happens in the stomach is this churning action, which is what leads to liquefaction. Now, I've always wanted to try this, and I tried it this morning with my other class, which is to demonstrate liquefaction. First off, the stomach, when it's empty, is rather small. It's about the size of this kind of Ziploc bag. But it can expand to about this size Ziploc bag when it's full. Okay? So I use this Ziploc bag to demonstrate liquefaction. So I took the other half of this hamburger bun and put it in the bag. And I broke it up as if it was being chewed up. Now, when you're chewing food, you're getting it to a kind of pasty consistency, but I was just putting dried bun in here. And then I did the churning within this bag. Now, actually, the stomach, like I talked about with Og, has sort of an opening at one end and an opening at the other end. I'm not going to open this thing because this is disgusting. But this would be the end that's going on into the small intestine. This would be the end that's receiving stuff from the esophagus. And what happens in the stomach is it churns the food back and forth kind of like this. Okay? And as it's doing that, it's mixing the food up in a water solution and liquefying the food. Okay? Now, this seemed like such a great idea when I did it, when I thought about it before doing it this morning. But even with sitting here and churning around the contents of this Ziploc bag for several minutes, and I've done it forever, it's not completely liquefied. You can see floating in here little bits of bread. So it's kind of disgusting. But this started out as clear water. And so the liquefaction process is really the bits of the food kind of dissolving into the water. And it'll be easier and easier for enzymes to get in there and start to break these things up. Okay. With that churning action, as the food is pushed towards the end of the small intestine, a little bit moves out of the stomach into the small intestine. And the rest of it just gets churned back the other way. So it just keeps going back and forth like this. And a little bit moves out of the stomach into the small intestines with each wave of churning. Now that churning action is sort of a more complex peristalsis. Waves of contraction move down the length of the stomach from one end to the other. And then when the food gets down here to the valve that connects to the small intestine, a little bit gets out, but most of it washes back the other direction and it churns the other way. And then another wave of peristalsis pushes it this direction. Most of it churns back the other direction. It just keeps going back and forth, and a little bit is introduced to the small intestine at a time. Now, I showed you the stomach has sort of a smaller size when it's empty and a bigger size when it's full. The stomach can stretch to accommodate volume. The small intestine can't. The small intestine can hold a great volume of food, but not all at once. So a little bit's introduced at a time, and the intestines are these long tubes. Is this long tube. And so a little bit moves into the tube at a time until the whole tube gets filled up with the contents of the stomach. And in the process of churning it back and forth and letting a little bit move on, what's still in the stomach gets liquefied in that process. Okay? Now, when I'm covering this stuff in a regular lecture course, usually I try to get it over 2 lectures. And the first lecture I get up to the stomach, and I don't quite finish the stomach. And so when I come back for the second lecture, I start by finishing up the stomach, and then I go on to the intestines. So with that in mind, I think this is a good time to stop because we're right at that kind of midway point. So we'll go ahead and take our 20-minute break, and I'll come back and start talking about the histology and more of the function of what's going on in the stomach and how it's chemically digesting food. So we'll pick up there with the rest of it. Now, this is really cool. Okay. I've switched to this new way of doing the capture. See how it's showing this infinite regress of the screen? Wow, cool. Okay.